So I'd like to begin by thanking you all for joining me this morning for this webinar. I hope firstly you all are keeping safe and maintaining social distancing rules as per the country where you're located. So the topic for today's webinar, as I mentioned earlier, is autoimmunity. And the focus of today's talk is on type 1 diabetes. So I worked on type 1 diabetes within the endocrinology therapeutic area for quite some time now. And today I'd like to talk about some of the results, discuss the results with you. So basically, for a more specific description of my talk today, I'll be talking about the role of hypoxia-inducible factors in islet transplantation and type 1 diabetes. So obviously, a big thank you to Amity University and also to Dr. Harsha, who I was in regular touch from the past few days over LinkedIn. I was just talking to her a little while ago, and I was saying to her, it's a really nice initiative that she's doing in terms of educating everyone during these unpredicted pandemic times. So with that in mind, I hope you all get some fruitful information after listening to this webinar today. So I just thought I'll begin the webinar by giving you a very quick background on what I've done, about my background, what I've done in the past, especially before I got into endocrinology research. So before I started my work in endocrinology, I worked as a project manager within the respiratory therapeutic area. So here are some of the publications that I contributed towards. And basically, our lab was involved in coming up with novel biologic therapies for asthma and COPD. So I had quite a nice, successful time during my research time in respiratory, where we came up with a successful biologic therapy that's currently in human trials at the moment. So I'd like to begin by talking a little bit about giving an introduction about autoimmunity. So what is autoimmunity? So in simple layman terms, it means that when your immune system has gone rogue against you, it leads to an autoimmune disease. So I'm sure we all know that our immune system is designed to protect ourselves against foreign invaders, such as bacteria, viruses, worms, etc. And it does this with the help of various immune cells that's shown on the slide, such as T cells, B cells, and larger macrophages, dendritic, and neutrophils. So these immune cells, obviously, as you know, they're also known as white blood cells. But in some individuals, their immune system thinks of their own self-cells and organs as foreign, and they mount an immune response against it, meaning they attack those cells and organs, and thus that leads to an autoimmune disease. So there are many reasons why this happens. So obviously for today's talk, in the interest of time, I won't talk about those various reasons, but I'll mainly talk about type 1 diabetes, which is also a very well-known autoimmune disease. So I'm sure you all know, but I'll just give a really quick background on type 1 diabetes. So it's a disease where your pancreatic beta cells are destroyed by your immune system, and hence your body is not able to produce insulin. So beta cells are the ones that produce insulin. So when they get destroyed by the immune cells of an individual in an autoimmune attack, they're not able to produce the insulin, and hence there is accumulation of glucose in the blood, and thus it leads to high blood sugars, and if uncured, it leads to many harmful complications. So just here in this animation, you can see the various immune cells that are involved in the autoimmune process in type 1 diabetes. They do this by release of various proteins and cytokines, thus leading to the destruction of the beta cells. So very quick, so we understand now what is type 1 diabetes, but how is it really caused? So just again, quick background on so the development of type 1 diabetes starts with an unfortunate concurrence of genetic susceptibility and an environmental trigger that causes an imbalance between the regulation and immunity that further leads to the onset of insulitis. And when about 10 to 30% of the beta cell mass remains in an individual, that's when it leads to hyperglycemia or high blood sugars, and hence clinical diagnosis of type 1 diabetes. So environmental triggers is a very well-known causal factor for type 1 diabetes, and it's actually quite a debatable topic for over 5 to 6 decades. And mainly it's debatable, I'd say, because a lot of the research data that's available on environmental triggers is not thorough. So basically, in my project, what I wanted to look at was one environmental trigger that's highly susceptible in causing type 1 diabetes. I wanted to study that environmental trigger. So basically, the environmental trigger that I studied is an enterovirus, and more specifically Coxsackie virus B4. So it's considered as a prime viral candidate for causing type 1 diabetes. It's quite small in size, and it's also been extracted and cultured from pancreases of type 1 diabetic patients that were recently diagnosed. So in terms of its structure, it's quite a small virus, about 30 nanometers in size, and consists of mainly four structural proteins known as VP1, 2, 3, and 4. So the first three proteins, they are located on the capsid outside of the virus, so known as a capsid, and the VP4 is located on the inside. And lastly, it consists of a single-stranded RNA genome, and it can infect, so it's got a high propensity of infecting pancreatic beta cells. And I'll just play a quick animation of how it does this. So initially, it can infect beta cells via the coxsackie adenovirus receptor, which is also known as CAR receptor. And once it's within the beta cells, translation of structural proteins and viral genome replication occurs in the Golgi and endoplasmic reticulum. Then the newly formed viral progeny, which is shown in number 4, they basically exit the beta cells via exocytosis, and it leads to beta cell death. And you can see that it leads to cell wall decimation, and hence it leads to beta cell death. And this further on leads to insulitis and potentially clinical type 1 diabetes. So what are the current therapies and prevention approaches for type 1? So obviously, I'm sure you know this. So the first one's insulin, so multiple daily injections. So this is done by the patient getting multiple daily injections, the normal traditional way with a syringe, or also there is an equipment called insulin pump that helps to achieve better glucose control. And the second is prevention approach, which is by controlling your diet. So the current cures for type 1 diabetes include whole pancreas transplantation, which is surgically quite invasive, and the less invasive is the second one, which is islet transplantation. However, islet transplantation is still experimental, and what I mean by that is that its success rate is not 100%, and hence more work is needed to be done in this area. So in spite of many approaches, including immunomodulation, all have failed to really address the issue or coming up with an efficient cure for type 1. And hence, on the good note, there's a lot of research that's been carried out in this area. But more recently, attention has turned towards beta cells. So more attention has turned towards beta cells in the development of type 1 diabetes, and specifically identifying genes and proteins within the beta cells that can actually lead to cause the disease. So you can imagine that if the beta cells are they are provoking the immune system to go against them and attack them to cause type 1 diabetes. So in order to investigate that, there are a lot of ways you could do that by identifying the different genes and proteins that are released by the beta cells during the autoimmune process that actually leads to cause of insulitis and hence diagnosis of type 1. So next up, I want to talk about this family of proteins that that I've worked on, and that's the topic of today's webinar. So the basic helix loop helix PAS superfamily of heterodimeric transcription factors are proteins that sense and respond to environmental or physiological signals. So each transcription factor in this superfamily consists of two PAS domains known as PAS A and PAS B. So as shown over here, PAS A, and so that's the PAS A, and this is the PAS B. Also a beta helix loop helix dimerization domain. And lastly, they're divided into class 1 and class 2 members. So the class 1s are on the top panel and the class 2 at the bottom. And basically, in order for these transcription factors to work efficiently, there has to be a binding between these two class members because each subclass member won't be able to work on its own. Hence, the binding is quite important. But for today's talk, I'm just going to talk about the importance of HIF1 alpha, which is in the top panel, which is a class 1 member, and the class 2, and its obligatory class 2 member is ARNT. And I'll talk about these two transcription factors in islet transplantation and in the cause of type 1 diabetes. So basically, under hypoxic conditions, so hypoxic conditions, I'm sure you know, are also known as low oxygen conditions. So HIF1 alpha binds with ARNT to form a dimer, and this helps in the transcription of various genes involved in the hypoxia response element. And hence, this is a way for a cell to come up with a protection strategy under conditions like hypoxia. So basically, when HIF1 alpha and ARNT bind together, a combination of these two factors, which is a dimer, they're together known as HIF1 or hypoxia-inducible factor 1. So for today's webinar, I'm just going to run through these few aims. But more importantly, I'll talk about the first three aims in more detail because that data is published already. And with regards to aim 4 and 5, I'll just give you a brief idea of what's been going on with those aims at the moment, because mainly it's unpublished and I can't talk much about it. So basically, the key aims of the projects were the first one to determine the role of beta cell ARNT. And obviously, I wanted to look at the function of this transcription factor in beta cell function, and more importantly, in islet transplantation. So more specifically, I wanted to look into ARNT is mainly because the lab that I was working in at the time, they had already published results on beta cell HIF1 alpha, which was involved in beta cell survival. But there was not much work done in the beta cell ARNT, so that's when I came in and I wanted to work on that. So, and the second aim then was to investigate the role of beta cell HIF1 alpha in the susceptibility of type 1 diabetes and or beta cell death after pancreatic insults or immunological insults such as Coxsackie viruses, strep rosetosin, and IL-1 beta. And last one is to study the effect of knockdown of beta cell HIF1 alpha in human islets. And this is followed by Coxsackie virus infection and also IL-1 beta stimulation. So the first two aims were preclinical models where all the work was done in animal models. And then the last one was more of a semi-clinical model where we worked with human samples and human islets. And the main idea behind that was to see if we can mimic the results that we had from the first two aims in human samples as well. So to begin with, so the first aim, like I said, was understanding the role of beta cell aunt in beta cell function. So like I said, our lab had published a paper already on HIF1 alpha in islet transplantation and looking at beta cell survival with this transcription factors. So basically, the take-home messages from this paper were that when the mice that lack beta cell specific HIF1 alpha, when they were compared it with their controls, they had increased apoptosis, decreased beta cell volume, decreased beta cell glucose tolerance, and due to that, the islet transplant success rate was quite decreased. So when I joined the lab, my role was to really understand what about the role of aunt in this whole process. So we know that HIF1 alpha plays a very important role, but does aunt also have a similar role in islet transplantation? So basically, to address that, my hypothesis and aim. So the first part of the project was to investigate beta cell function and islet transplant outcomes in islets lacking aunt. So basically, the study was designed in such a way that approximately 3 days prior to transplantation, a glucose tolerance test was performed in the donors, and then the recipients were made diabetic with a single injection of streptozotocin. And then once diabetes was confirmed, we did a transplantation. We ran the transplantation model from the donor mice in basically 3 sub models. So 3-to-1, 1-to-1, and half-to-1. So what that means is, so 3-to-1 means transplanting islets from 3 donors to 1 recipient, and then 1 donor to 1 recipient, and then half the islets from 1 donor to 1 recipient. And basically, we wanted to understand these 3 different transplant models is because the islet transplantation in humans is quite tricky. So the first thing is there's quite well, like all transplant studies and work, there's a scarcity of donors. So in order to achieve a successful islet transplant outcome, we had to come up with a certain model that really works. So we wanted to test which one of these models is more efficient. And basically, after that, we performed a glucose-stimulated insulin secretion test. So I believe that was, if I remember correctly, that was at day 14. And then finally, towards the end of the study, we would perform another glucose tolerance test at day 25, and then the final nephrectomy at day 28. So the nephrectomy is when you remove the kidney of the animal model. And basically, we perform this nephrectomy is because when we transplant the islets, they are transplanted under the kidney capsule. So because it's quite invasive, it's an invasive surgery, so we can't transplant islets back into the pancreas, so we do it under the kidney capsule. And this has been tested in various groups, various labs around the world, and it works really nice. So obviously now we had to perform a nephrectomy just to make sure that when we did the islet transplantation, did it actually work? And you do this by when you do the nephrectomy. If the mice start having high blood sugars, again, it means that the islets were working before, and because of the nephrectomy, the diabetes has reoccurred. So there's different experimental mice that I used for this aim. So they were mainly on a C57 Black 6 background. And basically, I produced these mice using the Cre-Lox recombinase system. So for those who are not aware of the Cre-Lox recombinase system, it's basically, in short, you have 2 LOXP sites on a certain region of your gene of your protein of interest. So in my case, it was the aunt, and this was done on the rat insulin promoter. So you can read a little bit more about Cre-Lox recombinase system if you want to obviously know more about it. So basically, so for controls, I used three different types of mice. The first one was flux control, so meaning where they only have the LOXP sites. The second was RIP-Cre control, so meaning when they only have the RIP rat insulin promoter gene in them. And then SCIDs were used as the recipients, and then finally our main experimental mice, the beta aunt mice or BANT mice, where the aunt gene has been specifically knocked down or knocked out from the beta cells. So here are the first set of results where the BANT recipients were glucose intolerant. So this was done before we performed the transplantation. So this is in uh, it's a comparison between donors and recipients. So you can see there's not much of a difference in terms of their glucose tolerance test. So which was a positive sign because obviously you don't want your donors to be glucose intolerant. Obviously, before they donate islets to the recipients, because if they are glucose intolerant, meaning they might have pre-diabetes, and obviously that's not a good sign if you want to give that to a recipient who's looking for a cure. Uh, so these are the results, the blood glucose levels, the random flat blood glucose levels from the first transplantation model, which is one donor to one recipient. So in the interest of time, I just thought I'd talk about the one model that I really had the most positive results with. So if you remember, I had mentioned I did three different models, 3-to-1, 1-to-1, and half-to-1. So 3-to-1 donor, 3-to-1 model was the most efficient model, which obviously is a no-brainer because you're having islets from three donors, which is a large amount of islets into one recipient. So they bound to work. But very interestingly, when I ran the 1-to-1 model, the results were quite interesting as well. So I'll just talk about the results and hopefully I'll convince you that these results, this model is quite efficient as well. So basically you can see from day 0 when the mice were very diabetic, when we performed the islet transplantation, the recipients started having reduced blood glucose levels. I guess the main point over here that I want to point is when we compare the BANT recipients with the control recipients, we can see there's a nice significant difference between the two groups of mice. And obviously, the control mice are expected to achieve normal glucose levels because they control and they're absolutely normal. But the beta aunt mice, obviously, they have a gene knocked out from the beta cells, so they're bound to perform abnormally. But I say these results were quite interesting because if you look at the blood sugar levels in the beta aunt recipients, they're quite normal. Well, not exactly normal, but very close to the normal ranges, I would say. And when we performed a glucose insulin secretion test, we saw that the insulin secretion in the beta aunt mice in the 1-to-1 model was significantly inappropriate. So what I mean by inappropriate is when we compare, so if you look at the blood glucose levels on the left graph over here, so the control mice are in blue and the BANT mice in red. So obviously the BANT mice have higher glucose levels when they were stimulated with glucose, so meaning that they're not able to produce enough insulin. And then when we compared it with the insulin secretion test over here, so there was not much of a difference in terms of the insulin secretion, the amount of insulin that they're secreting. So this raised the question in my mind that, okay, so if they are secreting the right amount of insulin, maybe it's a non-functional insulin. So meaning it's not able to perform its function efficiently, and that's why, in spite of having normal secretion of insulin, the BANT mice were still having high blood glucose levels. So with that in mind, I went ahead and enumerated the beta cell volume and did some histological analysis. So here we can see that the Barnt mice had an increased trend of the beta cell volume as compared to the flux controls. So the beta cell volume over here in the histological micrographs is shown in brown color. So you can see the control. So this is a histological section of a kidney. It's a kidney capsule. So basically that's where the islets are transplanted. So we can see in the control kidney graphs, the islets appear quite normal. But in beta, they're quite large in number. But you can see there is a lot of degranulation appearing. And what I mean by that is they're not condensed. They're quite loose. They appear quite loose. So that led to a conclusion that there might be a problem in terms of insulin function in these mice. So the next step was to enumerate some of the genes involved in various pathways. So obviously I went, so the normal enumeration was to look at different apoptotic pathway genes. So I looked at BCL2, BAX, and BCLXL. So we can see that the Barnt mice had significantly reduced levels of these. So meaning that ARNT is not really involved much in beta cell survival and also in glucose sensing for GAPDH and GLUT2. There was not much of a difference, but there was a much significant difference in proinsulin, which was very, very interesting because our hypothesis previously, a speculation that the Barnt mice have inappropriate insulin secretion and non-functional insulin. So that sort of indicated that sort of got confirmed when we ran this assay. And so basically, to cut the story short, in a nutshell, this paper was published in 2014. And basically what we showed is that when the recipient mice receive islets from donors that lack beta cell ARNT, there is no change in their apoptosis, so meaning there is no change in the survival, but there is decreased glucose tolerance, and the glucose metabolism is decreased. And obviously, this is due to inappropriate insulin secretion and signaling genes. So with that, basically, when the recipient mice received islets from donors that lacked HIF1 alpha or ARNT, the islet transplant success rates were reduced in both the models, but obviously due to different reasons. For HIF1 alpha, it was the survival, whereas for beta cell, for ARNT, it was function. So next up, so like I said, our lab's done a bit of work on HIF1 alpha, on HIF1 obviously in islet transplantation and type 2 diabetes. So these are just a few number of papers that our lab had produced. But the next question was, okay, so HIF1 alpha, what's the role of HIF1 alpha in type 1 diabetes? And I more specifically just concentrated on HIF1 alpha in type 1 diabetes is just for a simple reason because, like I mentioned before, HIF1 alpha is involved in beta cell survival. So type 1 diabetes is a disease where your beta cells are depleted by in an autoimmune process, so meaning beta cells die. So to understand the role of HIF1 alpha, which is an important survival factor in type 1 diabetes, made proper sense rather than looking for ARNT in type 1 diabetes, which is more of a functional transcriptional factor. So basically the two aims, aims 2 and 3 of the project were to investigate the role of HIF1 alpha in the onset of type 1 diabetes, and basically we used 3 different models for it, so all immunological insults. So the first one was with an in vivo model where the mice would be infected with Coxsackie viruses. So we used 2 different strains of viruses, B1 and B4. The second one was with multiple low-dose streptozotocin, which is also an in vivo model. And the last one is with IL-1 beta stimulation. So this is an in vitro model. So again, so the different mice models that are used for these aims. So these were on a NOD background as compared to for the Barnt model, which was on a C57 black background. So obviously NOD mice, I'm sure you'd know, is quite a well-known mouse model for studying type 1 diabetes. So similar principle with Cradox recombinase system. So 3 different controls we use, and obviously our experimental mice, the beta HIF and alpha NOD mice, when HIF and alpha gene is being knocked out from the beta cells specifically. So just before I begin talking about the results, I wanted to show the small result that I had performed in comparing. So initially, when I wanted to use these mouse models, so I wanted to make sure that I use a mouse model that is not very susceptible to diabetes. So meaning, so NOD mice, as you would know, they mimic the human situation of developing type 1 diabetes by natural causes. So developing autoimmune process, encephalitis, and type 1. So female NOD mice are quite well known to have this process quite well established and developing type 1. But males somehow don't have that. So they sort of diabetes resistant. And the reason I wanted the diabetes resistant mouse strain was because I wanted to make sure that when I carry out my in vivo or in vitro models in these mice, and if they become diabetic, I wanted to make sure that they actually became diabetic because of the insult that I gave them, and not because of their natural autoimmune process that they have. So like I said, so the female NOD mice have high propensity for developing type 1 as compared to males, but this is quite opposite in the humans. In the humans, males are more prone to developing type 1 as compared to the females. But anyways, I won't talk about that for today because that's going to be altogether another different webinar. So to proceed, so basically I did a encephalitis scoring a test between these two between 8 weeks males and females of 8 weeks. And 8 weeks is because that's the experimental age that I use for all my of the mice for all my experiments. So basically, I gave them an encephalitis score range starting from 0, meaning no encephalitis, to 4, meaning highly encephalitis. So you can see that the females had significantly higher encephalitis scores as compared to the males. So once that was confirmed, so all my experiments were performed with the male mice. And basically the timeline was after 7 days of acclimatization, so from day -7 to -1, at day 0, the mice would be infected with Coxsackie virus infection, so either B1 or B4, and the dose was 10 to the 5 plaque-forming units. And at day 7, which is the peak of the virus, would be a tissue harvest. Day 21, which is the clearance of the virus, we'll do another tissue harvest. And the main comparisons were between day 7 and day 21 to understand how the virus really works in these mice. And when you, in the absence of HIF and alpha from the beta cells, how does the virus is the virus more vigorous to these mice in the absence of HIF and alpha? And if the mice are able to clear, the mice that lack HIF and alpha, are they able to clear the virus at day 21 or not? So obviously the comparisons were between the controls and the Barnt mice at day 7 and day 21. And then also we did a long-term study where we infected the mice and then we kept them alive until day 105. And we basically wanted to see that if what sort of immunity do the mice get in the longer term? Do they get any immunity in the first instance? And if they do, how well are they protected? If we do a second, if they get reinfected with the same virus, are they much better protected, or they do worse? So basically, the first set of results shown over here. So this is just a Kaplan-Meier graph where basically I've shown that all the control mice at the top over here, so the NOD controls, flux controls, Rip-Cree, and also the uninfected beta HIF and alpha controls, they had no incidence of diabetes at all. But when I compare between Coxsackie B1 virus and Coxsackie B4, which is in green and red over here, you can see that these mice started to develop diabetes quite early on. So from starting from week 2 to week 5. So obviously, uh, so I guess initially when we ran these results, we had no plans to run the long-term study because what we were expecting in this was by week 5 or 6, all the mice would probably be dead. But that was obviously not the case. So hence, that's what we were interested in running the long-term study to see how well these mice perform. So you can see basically about 60% of the mice in Coxsackie B4 virus developed type 1, and there was about 40% of survival rate compared to 50% in the Coxsackie B1 model. And then we also ran a different model where we wanted to study inoculation of Coxsackie B4 virus orally. And I guess we wanted to do this is because to mimic the real human situation, because in humans, these viruses, well, they infect humans via oral route, and hence we infected these mice by oral gavage to see what sort of survival do these mice have. So again, had similar survival rates, about 50% of the mice survived. And then just for transparency, we also wanted to see are we able to obtain similar results in female mice. So if you remember before I said the female mice have naturally developed type 1 diabetes, female not mice. But what happens when you infect them with Coxsackie B4 virus? So as you can see in this graph, they go on to developing type 1 diabetes quite early on. So this is at about week 1 or so as compared to the males, which is a bit later on at week 2. So this was another way to confirm that the sex of the mice that we were using was the appropriate type, and also the age of the mice was appropriate. So when we had these results, we were quite excited because this was sort of one of the first beta cell model that I had produced, indicating that when there is lack of a certain beta cell gene. So if you remember in the first few slides I mentioned that a lot of attention has turned towards beta cells, so the various genes and proteins within the beta cells that lead to type 1 diabetes. So obviously HIF and alpha being a gene of the beta cells, when we had these results, we were quite excited because we sort of discovered a gene that belongs to the beta cells that leads to the cause of diabetes. But I guess the main question I had at this time was, okay, so the mice are getting diabetic, but I'm not sure if these mice are type 1 diabetic or type 2 diabetic. Because if you look at the symptoms of type 1 and type 2, they are very similar. They have high blood sugar levels just in mice as well. But I wasn't sure if these mice are getting autoimmune type 1 or not. And basically, to do that, to understand the autoimmune process, I performed adoptive transfer studies. So basically, I would isolate splenocytes from flux controls or the virally infected mice, and I would adoptively inject the splenocytes into not-skin mice and then look at the development of diabetes. So very, very interestingly, we saw that the adoptive transfer study showed that the virally induced diabetes is autoimmune. So as shown in the graph, the mice that had received splenocytes from mice infected with CVB1 in green and with CVB4 in red, they went on to develop autoimmune diabetes or type 1 diabetes at between week 6 and 7. So that was a very nice confirmatory assay that we performed. So next up, we wanted to see, okay, so what are the different, when we did the tissue harvest at day 7 and day 21, so what sort of features do we see in the mice? So on the So I do apologize if this image is a little bit gruesome for some of you. But basically what I wanted to show here was when you look at the control mice in the left over here as compared to the beta-hyphen alpha mice at day 7 post-infection. So this is just in Coxsackie B4 virus. We can see that there is a lot of calcification, so these white areas over here. So that's a classic manifestation of pancreatitis. So well, we were obviously interested in pancreatitis is because Coxsackie viruses are very well known to cause pancreatitis. So I'm sure you'd know. Pancreas is made up of two separate functions or two separate parts, exocrine pancreas and endocrine pancreas. So exocrine pancreas is involved in secreting various enzymes for digestive purposes, whereas the endocrine is involved in controlling your blood sugars by release of glucagon, insulin, etc. So when the virus infects the pancreas, it initially infects the exocrine pancreas, and then we can see that formation of calcification is a classic sign, which was a nice finding that we saw at day 7, which is the peak of the virus. And then we wanted to look at the size, the weight, and the size of the pancreas. So we can see that at day 7, the beta-hyphen-alpha knockout mice had reduced pancreas weight as compared to the controls, and at day 7 it has a decreased trend. And here are some pictures of showing the size of the virus. So we can see at day 7 in the knockout mice, the size is quite small as compared to the controls. And similarly at day 21, similar case. So next, we wanted to, so I ran a biochemical test to look at the amylase activity. And I guess the reason why I was interested in amylase was mainly because amylase is one of the digestive enzymes that's secreted by the exocrine pancreas. And obviously, if your pancreas has been destructed by the virus, to confirm if your model is working really well, to detect amylase in the serum is a nice assay to confirm if your model is working well or not. So we can see that at day 7, there was an increased trend of serum amylase activity as compared to controls, and similarly at day 21 as well, which is at the clearance. So next up, we performed a plaque assay. So we can see at day 7, the beta-hyphen-alpha knockout mice had more number of plaques as compared to the controls, and similarly at day 21, there were no plaques, which was quite expected because the virus is cleared. And also, based on the plaque assay, I did a viral load enumeration, and we can see that at day 7, the knockout mice had higher as compared to controls. And similarly, I also did a virus, so VP1, which is one of the capsid proteins of the virus. I did a stain of that, and we can see that at day 7, a lot of brown, meaning a lot of VP1 staining. So if you compare the controls with the knockout mice, we can see there's a lot in the knockout. And obviously at day 21, you can see that because of the infection of the virus, there's a lot of the pancreatic, exocrine pancreatic tissue is lost. So next up, I also did beta cell mass. Again, the knockout mice had, the uninfected mice had obviously normal beta cell mass as compared to at day 21, which is the clearance of the virus, they had much reduced beta cell mass. Then we've also interested in looking at fibrosis. So we just did a serous red stain where, again, we saw that the beta-hyphen-alpha mice the beta-hyphen-alpha transcription factor may have a role in pancreatic fibrosis. And this is shown by the deep red staining over here in these mice as compared to the controls. So next, obviously, I mentioned so type 1 diabetes is caused by increased insulitis, and we wanted to enumerate this insulitis. But I guess in the bigger picture, what we wanted to really do was look at the encephalitis and more specifically, I wanted to look into what are the different populations of uh, of the different cell types within this encephalitis population. Uh, so we can see in the knockout mice at day 7, so the islets are stained in brown, and you can see there's a nice collection of immune cells, encephalitis around the islet. So obviously the knockout mice had more encephalitis as compared to controls, and similar story in at day 21 as well. So when we went ahead and looked at when we counted these number of cells around the islets, so at day 7, there was increased number of inflammatory cells per millimeter square of the islet area. And at day 7, it was significantly increased as well as compared to the controls. So next up, as I mentioned before, I wanted to look into the different population of the cells. And to do this, I ran a FACS assay, so flow cytometry assay. So basically, over here in the plots on the left, you can see uh, forward scatter, side scatter plot. You can see in the knockout mice in the bottom panel, they have a higher number of lymphocytes as compared to the controls. And obviously, when we enumerated the specific cell types, we saw that the knockouts had increased CD3, CD8, and CD19 B cells. So this is just a microscopic graph or showing that what happens to the islets when they are treated with when they're infected with the Coxsackie B4 virus in a culture plate. So obviously, when it's within a mouse system, you can understand it's more the virus is more happier in a system because everything it gets all the nutrients and everything from a living organism. But obviously, when it's in an artificial in vitro plate system, it's a bit different story. But in spite of that, what we saw was comparing at 0 and 24 hours, we saw that when we knocked out beta-hyphen-alpha from the mouse islets using transfection model, we saw that these mice had refragmentation as compared to the control mice over here. So next up, the obvious things were to look into the genes that were involved in various different pathways. So I guess I might be running out of time. So just in the interest of time, very quickly, what I looked into was ER stress, oxidative and RNA sensors. So we can see that the HIF-1 alpha knockout mice after infection at different time points. So at 0, 4, 7, and 14 had. So I guess I just want to point out over here is that so these are from 4, 7, and 14 days post-infection. Obviously, I couldn't get many islets from day 21 because, like as you saw in the previous histological images, there were not many islets left. So meaning I wasn't able to extract islets from 21-day infected mice, and hence I had to go with 14-day infected mice. But still, we can see that they significantly increased. And similarly, we wanted to run a different model where we wanted to treat the mice before the infection with actinomycin D, which is basically a transcriptional inhibitor. And we wanted to use this model to see if we're able to inhibit the transcription of the virus and then somehow stop the aftermath of the virus effect. But what we saw was that in spite of that, the apoptotic and RNA sensors genes were quite highly elevated in the beta-hyphen-alpha mice as compared to their flux controls. So the next thing, so this is I'm going to go on to my third aim now, where we wanted to see that if we're able to replicate these results in human islets. So obviously all this work was done in preclinical mouse models. Now, if we're able to do this in human islets, that would be like sort of a holy grail achieved. So to do that, now when you do mouse models and preclinical models, knocking out a gene in a mouse or knocking out a gene in some cells from a mouse is quite easy. But obviously in humans, when we isolate islets from a human, they have normal levels of HIF1 alpha and other transcription factors. So to basically to mimic a situation, we had to really knock down HIF1 alpha gene from these islets. And to do that, we employed transfection model. So using lentivirus, so we can see that we achieved more than 80% of transfection using a GFP marker. And this is just a small time-lapse movie that shows what's the effect of Coxsackie B4 virus on human islets over 24 hours period. So basically 1 second of the movie is equal to 2 hours. So the movie went on for 12 seconds, so it's 24 hours. So you can see that when the mice when they had when HIF1 alpha was knocked out from the human islets and when they were treated with Coxsackie B4 infection, there is increased defragmentation. So I won't talk much about this one because that's a different topic altogether. And again, I won't have too much time to talk about it. But basically, these were the results that we saw upon infecting the human islets with Coxsackie B4 virus. And then we also ran some real-time PCR to look at to enumerate the viral genes within these islets, and we can see that when there is no HIF1 alpha in the human islets, there was increased amount of these Coxsackie virus B4 genes. So it's basically we trying to quantify the 5' untranslated region from the Coxsackie virus. And then we obviously ran some more PCR studies where we saw different levels of different genes. So the first one is obviously the control islets, where basically the mice had, so the human islets had proper amounts of HIF1 alpha, and they were not infected with Coxsackie B4 infection, and then they were infected, and then they were infected in the presence of HIF1 alpha, and then this was another negative control, and this was infected in the absence of HIF1 alpha. So you can see there's a nice trend of increased levels of RNA sensors, increased levels of apoptotic genes amongst even in human islets. So basically what I can conclude from these results is that we were successfully able to mimic what we found in the human islets. So the second model that I wanted to run was on multiple low-dose streptozotocin. So it's a similar model that as compared to the Coxsackie viruses. So here's the model. And basically, here are the results where we saw that when mice, beta-HIF1 mice, after multiple low-dose streptozotocin, had reduced weights and increased blood glucose levels. Similar story in the glucose tolerance test in all groups of mice, and also they had increased incidence of diabetes, and the mice were dying because of the infection, because of the streptozotocin treatment in the absence of HIF1 alpha. Again, and these are just the histological images where we looked at the beta cell mass. So obviously the ones treated with MLDS, the beta-HIF1 alpha mice had reduced beta cell mass. And then the last model that I ran was the IL-1 beta stimulation experimental model, which was an in vitro model. And basically what we wanted to see was what's the effect of IL-1 beta on control islets versus islets that are knocked down from beta-HIF1 alpha mice. So we can see here, so I ran 4 different time points, 0, 1, 2, and 24. And we can see at 24 hours, more specifically, a lot of the islets are lost. So you can see there's a loss of islets in the media, the control mice as well. But when you compare it with the mice that were infected with the virus, there is a much significant loss, so which was also a very good finding for us. And then we ran some real-time PCRs for anti-apoptotic, for pro-apoptotic, and for beta cell survival genes. So again, the beta-HIF1 alpha knockout mice islets that were treated with IL-1 beta had decreased levels of anti-apoptotic, increased levels of pro-apoptotic, and decreased levels of beta cell survival and maturation gene. So similarly, we did similar studies in human islets as well, where we knocked down where we treated human islets with the with the IL-1 beta cytokine, and similar story, we had decreased levels of anti-apoptotic genes as compared to increased levels of pro-apoptotic. So with that, I'll sort of end today's webinar. So all the results that I just showed you have already been published in the paper here on the slide. So feel free to have a read of the paper. It's an open access journal, so it's freely available to read.
Yeah, the first question is from Dr. Nimisha Srivastav, and she's asking regarding pathogenesis of type 1 diabetes diseases in humans, whether the infiltrating inflammatory cells themselves express cytokines that lead to cellular cytotoxicity, or it is because of autoantibody formation. Please elaborate.
Okay, so well, thanks for the nice question. So I guess to answer it more specifically, I guess what I understand from the question is that whether the insulitis, whether the immune cells within the insulitis, they have increased, do they release increased levels of cytokines? Yes or no? So I would say yes, and we've already sort of checked that in my model. So I wouldn't really say it's beta cell autoantibodies. Because obviously cytokines and autoantibodies are two different things. Cytokines would be more present in the insulitic areas, which leads to, with the release of chemoattractants and cytokines, there are other cells that are recruited towards that infected site, and hence. So my answer would be it's more cytokines as compared to autoantibodies.
And the second question is from Dr. Subhash Chandra, and he's asking, he's thanking you for this informative talk.
No
and
problem.
he's asking, my query is that can we expect stem cell therapy as possible treatment of type 1 diabetes in near future?
I would say yes. So stem cell therapy work has been going on with a lot of groups are already working on it at the moment. But I guess the same problem is stem cells. It's about the immunological problems, right? So when you have stem cells, when you transplant stem cells into a diabetic recipient, there's always they considered as foreign, and then they mount a very vigorous immune response against it. So a lot of immunomodulation techniques have been applied to achieve success in this area. So there's a lot of research going on at the moment. But I guess, I mean, if you want to compare stem cells to sort of in the model that I just described, where your beta cells don't have HIF and alpha. So I also performed many studies where I increased the levels of HIF and alpha, and then I saw what happens in terms of diabetes incidence. And the results were positive, of course. Now, so these aren't published, so I won't talk much about it. But basically, there is a nice scope with stem cells. So if you can have stem cells from a normal individual and then have increased HIF and alpha levels in the diabetic recipient, so you can pretty much achieve good success with that.